So, good morning, good afternoon, everyone. So, I will tell you about the in vivo dynamics of RAD51. So, luckily, you already had a very good introduction this morning by Heiko Vanatikum. So, basically, double-strand breaks are really a dangerous events for the cell, for the genome. If they are not repaired, they can lead to cell death or gross chromosomal rearrangement. And there are different pathways, as we heard this morning, to fix them. And the one I would like to discuss with you today is homologous recombination that is initiated by the resection of the two sides of the double-strand break, leading to a single-strand DNA that will be covered by RPA, as Heiko mentioned. And then in budding yeast, it's RAD52, which will load the recombinase RAD51. So RAD52 does the job of BRCA2 in human cells. Once the filament is covered by RAD51, actually this is the nucleofilament, and this is a physical object that will undergo the search for an homologous sequence, and that will be able to invade it. After this invasion, you will copy the missing information and then fix the problem. So this is a textbook picture, which is quite well established, and it works super well on the textbook because the homologous sequence is always just below the double-strand break. So this is in the textbook. And actually, it's probably also true in the cell. If it is one of the sister chromatids that is broken, you can use the other sister as a donor. It's a perfect donor. It has a perfect homology, and it is probably in very close proximity. But what if it is not present in the nucleus? Then you have to look for a distant homologous sequence, which in budding yeast will be provided by the homologous chromosome. So we heard that in marmots it will be very far away. But in budding yeast it works pretty well. And the big question we wanted to address is how do you reach this donor sequence among the megabases that are present in the nucleus? So actually you are down to the very old question, how to find a needle in a haystack, so for which you have different strategies that have been proposed. So here are a few examples of it. But what we know from biochemical studies, actually, is that this object that is undergoing the search is this RAD51 filament. So it is forming a very stiff structure in the, as you can see here in this electron micrograph grid. It doesn't really work, this thing, but it's okay. So it makes a very stiff filament, and it's important to have in mind some numbers, actually. So it's really stretching the single-strand DNA up to 1.5-fold the B form of DNA, meaning that if you have 1 kb of single-strand, you expect 500 nanometer of RAD51 filament. And as we also heard this morning, you can have quite extensive resection in the cell. So it is estimated to be up to 4 kb per hour, the resection rate. So you expect quite few micron long filaments. But actually, we could not really... thank you. So this is for the in vitro, and there is beautiful work on single molecule in vitro studying the dynamic of this process, but we don't know much in vivo for the reason that actually so far there was no RAD51 tagged functional that were available. But what people had done in yeast is that they introduced a tag in the C or N terminus of RAD51, and what they could observe was a focus, not at all this very long filament. But as I already mentioned, these were not functional, and it's not surprising if you look at this super nice cryo-EM structure of the filament, you can see that it's going to be hard to put a tag on it without impacting on the structure. But yet, we thought, okay, let's try to tag it. And so, yeah, one more comment. So people, when they were observing this, they were a bit puzzled, but they say, okay, but anyways, all repair protein from foci, and maybe in the living cell, you don't have a continuous filament, it's full of little gaps, which allows the filament to kick, and you can have the filament folding into this focus. This is why, this is how the things were interpreted so far. And actually, it is true that if you look at if you look at other repair protein, they all form this nice focus when you induce a double-strand break. So this is RFA1, which is RP1, RPA, sorry, and RAD52 is also forming this focus. They will colocalize. And so just would like to briefly mention this study that we published 2 years ago with Judith Mineatab. If you do a single molecule tracking of these individual proteins, although they look super similar by a regular microscopy forming this foci, they show a very different dynamics. And I think it's really interesting, but you can look at the paper if you're interested. Okay, back to 51. So we wanted to tag it and to be able to visualize it. So we knew it was not possible to put the tag in N or C terminus. So we asked some advices from a structuralist, Raphaël Guérois. And actually, it advised us to introduce the tag in the N terminus of the protein, which is less structured. And he picked the place where it was the least conserved across species, with the reasoning that this is probably the place in the protein where you can make some changes. So very briefly, we characterize this tag. So here it is introduced in the genome. It is the only copy that is expressed. And if you compare, so here in this MMS sensitivity assay, you can see that the original N-terminal tag was really sensitive to MMS. Our first version of the tag was already much better. So we tweaked a little bit the system and we obtained a fully functional version of the protein. So, and we demonstrated that it's functional during meiosis and fully functional for gene conversion. So now we can look at them in living cell. So we use this system where we induce a single double-strand break at a specific locus in the genome by switching the cells in galactose. And by 2 hours of induction, most cells will have experienced this double-strand break. So at time 0, without any induction, we see from time to time, like a few percent of the cells is focus, which is probably due to spontaneous damages. After 1 hour, you see more of them. 2 hours, a bit more of them, but you start to see elongated structures. And at 3 and 4 hours, you really see very long structures, which we call filaments. So importantly, we checked whether this structure were also occurring in untagged strain, so using an antibody against RAD51 in the untagged strain doing immunofluorescence, and you can see that we also observed this long filament in the untagged strain. So actually, this is really reminding what people have observed in bacteria looking at RAK fused to GFP, forming this so-called bundled or filament. But in their case, it was not clear whether it was DNA associated or not. I mean, it's a different story, but it resembles. So a few controls. So we check that you need to have this long single-strand DNA to form this filament. So it has been introduced by Sahan this morning. If you delete both SGS1 and EXO1, you will have very limited resection, a little patch of single strand, and in this case, you can see that we don't form the filament. So the filament is really requiring the single strand, very extended single strand DNA. So we can follow the appearance of this structure over time. You can see that by 4 hours, half of the cells have filaments. So far, what I showed you was in a situation where you could not repair because we were in haploid cell, introduce a single double-strand break, no donor sequence, no homologous sequence. But yet we are focusing our observation up to 4 hours, which is more or less the time where if you can repair, you will start to repair. So I think it's still a relevant timing, but we wanted to look in a situation where you can repair. So here we are in a diploid cell. So we have an homologous chromosome that is a perfect donor. And we put a tag on it to be able to visualize the filament together with a donor sequence in the same cell and see whether the filament can contact the donor. As a control, we put a tag on another chromosome that doesn't have any homology. And we quantify the number of the frequency of the filament contacting the locus versus the frequency of the focus contacting the locus, the donor locus. And you can see that by 2 hours, half of the filament are contacting the donor sequence. And we have no specific contact with the focus. And it's interesting because by 2 hours, it's exactly the time point where we really see a difference in terms of the amount of structure between the situation when we can repair or when we cannot repair. You see that in the diploid, by 2 hours, you are much below what happened in the haploid. So in the haploid, it will stay forever, while the structure will peak at 4 hours in the diploid and then decrease. So these structures are really disappearing after repair, and we can see the contact at the timing of the repair. So now that we were convinced that these were functional structures, we wanted to analyze them a bit in more detail, so we decided to measure their lengths, because it was quite surprising as an observation. And you can see that, so here is the distribution of lengths over time. So you can see that by 4 hours, the median length is 1.3 micron, which could seem long, but actually, as I mentioned at the beginning, it is not that long, because 1.3 micrometer will correspond to 2.6 kb of resection, which is exactly what you expect. And we could also compare this with chromatin IP against RAD51, and you can see that this is cheap according to the distance to the double-strand break. And you can see that even this very long filament, that is 2.6 micrometer, which corresponds to 5.2 kb, you can see that at 5 kb you have a lot of 51 signal. So it makes sense, actually. It corresponds to the coverage of 51 along the DNA. So it's not only that we see filament, we also see kind of a zoo of structures, so we decided to classify them. So it's kind of an arbitrary classification, but so we have this very simple structure like the rods or the bent rods, but we also observe quite often actually some circles or some lasso that we put in the same category. And we also have these one node structures where you see like these branches. So we see up to 4 branches in this structure, and we have a kind of a mixed pot where we put all the others that are much more complex. And as you can see here, comparing the distribution of this shape in haploid and diploid, all these structures they exist in both cases. So these are not dead-end points; they exist in the diploid case when everything will be repaired at the end. So more complex structures are a bit less abundant in the diploid case. So maybe these ones are not that functional. So maybe you're surprised to see such complex structures out of one filament. But actually, if you think a bit more about it, you don't expect one single strand. You expect up to 4 single strands in a given cell, given that you have the 2 sides of the break, plus you have the 2 sisters. So you have up to 4 nucleofilaments. So I'm trying to distinguish the nucleofilament, which is a single strand covered by 51, from the filament, which is the thing that we observe under the microscope. So the filament is made of up to 4 NPF, but we see only 1 filament. So it is published for a long time that the 2 ends of the break are held together after the break. So maybe this is why we see just 1 filament, and this is usually the way people represent the 2 sides of the break in the textbook picture. Actually, we'd like to propose another organization. So we have a lot of argument about it, and I can discuss this further if you want during the questions. But we propose that rather than an end-to-end organization, is more a side-by-side organization, because this could account for many of the observations that we have. As you can see here, very often we have heterogeneous filament with a very bright part and a less bright part, and we interpret this as an asynchrony of resection between the different filaments, nucleofilaments, sorry. And also, I think this can also explain the one-node branch structures that have up to 4 branches. As you can see in this drawing, I think it's quite simple to imagine that if you separate the branch, you will end up having this structure. And actually, we even think that this separate structure could be related to the association of the individual filament with different parts of the genome. Also, in this situation, we don't have a donor. We can have microhomology, and this is what the search process is about, scanning for microhomology, and then, if it works, extending the interaction. So, actually, a good argument for this hypothesis is that in the RAD54 mutant, so RAD54 is essential for the invasion of the double-strand DNA and for the recognition of the homologous sequence. So in this mutant, we never see any branch structures. Instead, we see more circles. So what we think is going on is that when you have very long structure, at one point they will either branch or form circles. So I think these are 2 structures in competition. Okay, this was for the steady state images. So The fun part of it is that you can do movies, and so you can watch them in living cells. So this is a movie that is taken quite late in the process when the filament is already very long. And what is interesting here is that the filament is very dynamic, as you can see. And in this snapshot of the movies, what is really interesting is that actually you can see that we observe all kinds of shapes in a given movie. So it's not like each of these shapes is associated with a given cell, but it is very dynamic and alternating between these different shapes. So you can see circles here, you can see branches here, you can see like spiral. So this is a rare event, I have to say. Also, it's really striking. So we are always like catched by this one. So you can also do movie a bit earlier on, and so here this is what we did. You can follow the appearance of the first focus that will then extend. So this is an observation that we don't understand yet, but you start by a focus, and then you have kind of a cloudy structure that will at one point form a real filament that is really bright and straight. Another observation, so you can really, we can follow the dynamic of this extension, we can monitor how long it takes, we can compare in different mutants, so we have a lot of measurement about it. I'd be happy to discuss it during the question. What is also very striking is that so you have this very long straight filament that all of a sudden, from one frame to another, change totally its shape to seemingly collapse into a very bright focus. So we are really struck by this observation. So we decided to follow this a bit in more details. So this is another type of movies where we start, the filament is already elongated. Images are taken every 2 minutes. And you can see several of these events. I think it's pretty obvious here. You see the long filament and bright focus for a few frames that it will elongate again and again forming a bright dot here and there. So we can monitor this in the many cells. Actually, we see this event in average every 18 minutes. And so we thought, okay, what was striking is the change of shape and the change in intensity. So we monitor both. So here the blue line is the intensity over time. So this I didn't say, but these are 3D images. So this is the intensity over time. You can see variation, but much less variation than you observe in the 3D length. And clearly you have like abrupt change in lengths that are not associated with change in intensity. So we interpret this as this filament folding on itself, forming a focus. Okay, so we are really interested by this, so we decided to do a bit higher temporal resolution movie. So this is work from Fatma Lakal. I hope you had a chance to see her poster yesterday, but she's here. You can talk with her. So what so these events at high temporal resolution are hard to catch because we see them only every 20 minutes. But she could catch a few of them, and what she could find actually is that it takes less than a minute to transition from this structure to this structure. I'm almost done. Okay, interestingly, if you deplete cells, so the big question was, is this an active process or is it a passive process? So we decided to deplete cells from ATP, so it's a bit harsh, but just to see what happened. And actually, in 30 minutes, you transition from this long filament to this very bright foci upon ATP depletion. And she managed also to monitor this under the microscope, and you can see that in 2 minutes, you go from 2 minutes after adding the drugs, so it's very fast. You transition from the long filament to the focus. So this is very consistent with single molecular data obtained in vitro. So just wanted to also mention this mutant, which is a kinase ATPase dead RAD51, not kinase dead, sorry, that is unable to form the filament. So clearly this is an ATP-dependent process. So just to summarize, so we have developed and characterized a functional tag version of RAD51. They are very dynamic, and so the big question then is this dynamic important for the homology search, which was the initial question. And actually we think that because the filament is so dynamic, it gives a very nice way to explore the entire volume of the nucleus. So just to give you a feeling about it, so I just drew a line red line along the filament for each of these frames. And if you put them all together, for these 40 frames, you can see how much, how many directions have been explored during this time period. So you can see that it's really a very good exploratory engine. So this was our intuition, but we wanted to have a feel of the how much it helps. So for this, actually, we decided to ask some help from Leonid Mirny to have a bit more of to put some number of these different models and to know how much it helps to have a filament versus exploring the nucleus as a focus. This was the whole view of homology search, using having this little focus exploring the nucleus. So now clearly we see this extended filament, which is clearly exploring a much bigger part of the nucleus. And on top of it, we have this extension retraction, compaction extension phenomena that is also allowing the filament to explore different directions. So the question was, how do you find, I mean, how long it will take for the locus to find the donor in these three models? So for technical question, we will ask Leonid. But just the take home is that model one, so he's been able to, I mean, to derive analytical models and simulation, different type of simulation, but they all point toward the same conclusion, which is that, so if you look at the time it takes for cells to find the donor, so here is a fraction of cells that have found the donor over time. You can see that model one is working. It's not that it's not working, but by 12 hours, so after checkpoint adaptation, so thanks to Sahan, you heard this morning that after a certain amount of time, the cell will give up trying to repair and go through the cell cycle. So the aim for the cell is that what you want is all the cells having found their target by this time. So it's interesting to see that with model one, half of the cell only will find the target by this time, while with model 3, which is very robust and efficient, all cells will find the target, model 2 being in between. So I'm okay with the time, or? No, okay, I wrap up. So I just want to thank the people who did the work. So it was mainly the work of Siyu Liu with the help of Marie Wilmer and Judith Miniatab. Of course, I mentioned Fadma and our collaborators, Leonid and Raphael. Thank you.
That was really great talk. And if I can, so my question actually has to do with the issue that you are displacing nucleosomes over that range of where the Rad51 filament exists. And I guess what I'm curious about is, in principle, you have to reestablish the epigenetic identity of those nucleosomes after the double-strand break is reestablished. And it's kind of interesting that it's on the order of, say, like, 2 kilobases or so that the filaments exist, because that's, on the order of, say, 10 nucleosomes that is kind of reparable, right? Like if you look at what Leonid was talking about yesterday, the hotspots sort of propagate epigenetic identity over, like, say, 10 nucleosomes or so, and then that's as far as it's sort of reaches. So I'm curious, has you or has anybody looked at what the epigenetic identity is of those nucleosomes post double-strand break? And is there a dynamic response that, sort of, how this ties into reestablishing the healthy identity of that sequence?
Well, it's a very interesting question. We never approached it, so I cannot answer from my side. I mean, I'm sure there is some work about it, but I don't have it in the top of my mind right now. So
resections are short in mammals.
No, I don't think resections are shorter in mammals. They do very little homologous recombination anyway. So, and if they do it with a sister. In this case, you have much less. You have much less resection because you don't.
But it could be that there's like an optimization where you don't want the filament too long. You don't want the filament too long because, like, you could never correct the epigenetic identity, but you want it to be long enough that it actually aids the search process.
No, there is no. I mean, from what I know, in yeast it is very clear. I don't know if it is that clear in mammals, but there is no limit to the resection. It will go forever until because your priority is not to restore your epigenetic, it's to restore your DNA first. So and what is good with this process, what is nice with this mechanism is that the longer you wait, the longer the filament will be. And if you have to wait long, it means that the donor was not close. So I think it's quite interesting because you make this long structure when you need to make this long structure, when you need to search for something that is very far away.
Hi, fascinating talk. I was wondering, have you tried making multiple breaks and see how that happens and whether they collapse together or how they behave?
No, we didn't do it. I mean, we have indication that they probably go together, but yeah, no, it's on the list.
Very beautiful talk. I have a whole set of questions, most of which I'll leave for the coffee break. But maybe one that could be interesting also for other people is the regulation of this. So I think you are you're not controlling the cell cycle. You are inducing, as far as I understood your experimental setup, you're just inducing the endonuclease, and then they start arresting, and that's where they are. If you could have a G1 arrest and then induce the break, or if you watch this even longer, so into adaptation and then into the next G1, does this start changing?
Okay, so we did synchronize cells. So in G1, of course, you never form the filament. And when you release them from G1, you will form the filament right away. And it will resemble, I mean, in terms of behavior, it will be more or less the same, although we didn't study it extensively. And if you leave them forever, actually we didn't follow them forever, but we looked at them 24 hours after break induction, and we still see a lot of filament. It's crazy. I mean, it's like the cell is totally filled, I mean, the nucleus is filled with 51 structures.
So during adaptation into the next G1 start, it's just like it's dancing around this thing the entire time?
I didn't look at the moment they go through mitosis with the filament, so I cannot answer. We did it in checkpoint mutant, though, and I mean, we can discuss this. We have weird observation that I don't understand yet, but yeah.
Okay, okay.
Hey, that was very fascinating. One question I had is, you see all these different shapes, and I don't know if it's possible, but have you tried seeing how long each of these states last and what their transition possibilities are to different states to determine maybe whether this is some kind of common pattern that can help in the search? Or
Interesting. No, we didn't do that. But most of the structures are this simple structure actually. So yeah, we need to do many movies to really see all this transition and make sense out of them. But yeah, it's a good idea.
Do you have observed in your experiments and observation reposition of the whole chromosome during the early stages of homology recombination?
But we are not following the entire chromosome. We follow just the filament. So I cannot answer this question. But observation from others, I'm trying to think about it.
I think that the chromosome could be also moved to reach proximity.
Yeah. Okay. What has been?
Another process.
You're right, but
could be possible.
I don't know. We have to ask modelers, but I don't think it will be that easy to move an entire chromosome. It seems much simpler to have this filament that can extend like this, like a telescopic thing. But
yeah, Angela, have you looked whether these cycles of contraction extension can be modulated by factors that act on the filament, like SRS2 or like RAT51 paralogs?
Yeah, it's a very good question. So we tried, but we didn't identify any yet. I mean, so to answer your question, SRS2 doesn't do much on this. It does something on the structure and the brightness of the filament. 54 doesn't do much neither. Also, it affects also the brightness of the filament. Paralogs, they are really impaired in filament formation, which is interesting per se, but they don't form nice filaments.
I was a little struck in the models that you're reporting that like the rescue rate is twice as high when filaments are present rather than sort of a qualitative change, right? Orders of magnitude differences. And it makes me wonder, do you think this filament mechanism is specific to particular species? Do you think it's absolutely necessary for double-strand break repair, or is it possible that some species that happen to have a nice profile of genome size and rare double-strand breaks, et cetera, could survive without it?
I don't think I think they will all species will form this filament, maybe not with the same rate. Clearly, I mean, the fact that we don't have such a big of an effect, I think it's because the nucleus is very small. So it's still possible for the locus to find a target that is far away, given the size. In a bigger genome, I think it will be more important to be able to extend the filament. But it's true that in big genomes, the frequency of homologous recombination is much less if you give a distant target. So, yeah, I don't know. I mean, we are trying to tag it in other species, so we'll see.
Is it possible to reproduce these results in vitro, just to make a complex with the ssDNA, and after that try to see what is happening and to see whether there is a dynamics? Because you have an active kinase, right? And this depends on the activity of the kinase. So you should have maybe some other proteins which are associated, which are in a way associated or determining, I mean, the movement that you see, these fluctuations that you see.
Okay.
And is it possible to do, in principle, biochemistry, just to isolate it then, to sequence the complex, and to shed light on the mechanism?
It is possible. It has been done, actually, partly, not asking this exact question, but there are observations by the Green Lab using single molecule in vitro, and they show that if you just put 51, you will see this very nice elongated structure in the presence of ATP. If you wash out ATP, you will see kind of collapse. I mean, the filament will shrink. So it resembles very much what we see, actually.